Let me introduce myself. I'm an associate professor in the Division of Nephrology in the section of Quantitative Health in Department of Medicine, and I work on computation like machine learning, AI, or computational image analysis. And today I'm going to talk about something which should be of interest for all of you. I mean, at least somewhere or other is segmentation of kidney structures in digital pathology images, because the area that I work on is called digital and computational pathology. Ideally, I should have started this with a, I'm also affiliated with other department, but that's not important for today's talk. So ideally, I should have started with a real problem that the nephrologist might be interested in, that I was told one day, like 2 out of 15 diabetic patients are always in a problem. Yes, we're working on that, but let's step back a bit, because when people look at us, computers, like persons working on, looking at structures or mining data, they think that people can do magic and they can find out some some unknown pattern or something which should have significance, right? I mean, that's what people expect. But how many of those are in clinical practice? Because at the end of the day, the trustability is important, right? One of such things here that you can see, right? This is from a diabetic mouse with minimal or no proteinuria. And one thing you can see, like, forgive me my clinical knowledge, this is what I was told by my mentor, so possibly this is right, but here I cannot make a mistake. I'm in engineering department. So these are the podocytes. These are like coming on top of the capillaries, and they're almost trying to leave, right? But this guy doesn't. These mouse didn't have any proteinuria, right? So when people think about us, right, and even in current clinical practice, maybe people count number of podocytes, but they don't see like the angle and all sort of intrinsic detail to find out, if we can mine that data, to find out that thing, right, that automated, like, nice, interesting pattern, right, to what is hidden there. So that's the goal. Eventually, that's the goal. That's what we want to do theoretically. Trustability is important. Like, I forgot your name. Asudos, like Dr. Asudos, I mean, you would not even use our thing, anything in his clinic, because of trustability is important, right? So we have to first convince, right, that we can do something which could mimic like what you at least know, right? So even forget about, that finding about that pattern, right? So that's what I wanted to motivate. And at the end of the day, right, we all possibly either way or other, right, a common thing that we want to know: cell tissue interaction, what type of cells are there, right? What are their states, right? From normal states to abnormal states, happy tubule to little unhappy tubule, and the degree of unhappiness, right, in a continuum. And all these things we want to learn, or we want to learn, we want to learn, right? If Nemo is, there hidden, to find out that thing, right, using machine learning. But in order to generate that trustability, to make a big clinical impact, right, possibly we have to step back. So let me introduce a little bit of the digital concept of digital pathology, which you all possibly know. So I'll go a little fast in those slides. So what happens, when nephrology is the ordered biopsy, the biopsy is taken. It's not always needle biopsy, by the way, but it's been fixed, block is prepared, sectioned, put that on top of the glass slide, and then doctors manually observe this underneath the microscope, right, to write some charts like number of glomerulus, fibrosis, and all these things. But this thing is still manual if we are not using digital method. I don't think that you have done digital. I don't think so. But if this is a manual process, this whole process is time consuming. You can all sort of thing, like throw on that, like it is time consuming, laborious, it could have error, and so on and so forth. So that's why we bring computer, like someone like me, right, could be important, not important, like could be useful to help or contribute, right? We are in an era today that, this concept of digital pathology, where we no longer look at this small clock. If we are talking about computer, we have a whole slide scanner which can scan this whole slide image, which could be gigapixel size, which is very, very large. When I was doing my PhD, that time this kind of image, 1,000 whatever 32 by 1032 pixel image, that was considered to be a big data, like in 2003 or 4. But now we have like gigapixel size images, very large data. On top of that, we have, the advancement in other technologies, photonics technologies and spatial biology technology. For example, this is a Codex data. These are like 40 different markers showing different structures. And these gigapixel size images are bringing us opportunity and bringing us in the domain, in medical domain, to see if we can mine this data by bringing some tool to develop, bring that precision, right? And we take the help of, so thanks to all this, the development in computer hardware, do you know how to make this disappear? There should be a way to make this disappear. Push it up or yeah, here you go. So that computer hardware technology is rapidly booming, right? And then there is so-called machine learning, right? Deep neural network. So we can use all this using, using all type of new like large data. We can solve some basics problem first to generate the trust. And what are the basic thing when we try to look at, say someone says, oh, there is neovascularization in kidney, and someone doesn't know what is neovascularization. First, you want to identify the structure, right? What is it, right? In this case, this is glomerulus, of course, right? Which typically in engineering, so you don't go beyond glomerulus. Then you have, once you know what this thing is, you segment it. And then once you segment it, then you go to a precise question if there is like some sort of classification, what thing is happening, right? And by the way, this is a fake image, right? This is the last one, right? This is the area of the era of generating image from computer. So this image never exists, but this is computer generated image. But we don't need to go at that level of sophistication. We can only stick to like detection, segmentation, and classification. And the area I'm talking about, which is digital pathology, right, and computational pathology, it's booming, it's emerging, right? There are so many different scanning companies out there. Each time I go to conference, like, they're bringing with new products, new things, companies who are working in this area. The market is growing, right? And at the same time, there is new thing called spatial biology. We're talking about a grad program, by the way, AI for spatial biology for College of Medicine. And the co-director for this whole curriculum preparation. But now we're like this area is rapidly, every day there is a new technique coming, right? So basically this can measure like spatially different transcripts, different type of, proteomics, protein information, or transcription proteins. For example, this is a spatial transcriptomics, right? It's basically spatially resolved, like transcript information. Then this is codex. 40 different markers can be marked. This is called Cosmex data, and this is like something called 10x xenium, right? It goes even at cellular level, right? To measure the transcriptomics information. But that being said, right, I just wanted to gauge my audience. How many of you, right, in your practice, go to molecular level for making some sort of assumption? And that would might be a very stupid question, because I know I have few friends or collaborators with whom I work, but I just wanted to know, right, how many of you really go to the spatial molecular information and things like that and look at maybe I try to understand steps. Any of you use that in nephrology practice?
So for some things we do go at a molecular level, but some more broad structures we don't. So depending on what is our level of confidence in terms of clinical decision making, it depends. But
this is great point. So, but you see the technology is moving, because every time you go to a conference, right, nephrology, ASN or conference, they're like huge number of booths. They're coming up, hey, we can spatially resolve this many stuff. And now they're trying to push it to single cell level and for FFPE tissue, right? So all this data are getting generated, an amount of data getting generated, right? If I try to get all this data in our hypergetter, it costs me lots of money, right? I mean, then why this data is getting generated? When you see like, when your answer is it depends, right? So where things are moving, how we can merge it, how we can bring it to clinic, right? Then and only then we can all talk together and we can do things, right? To do something useful, right? So in order to get there, why I would still say there is a literature abundance. If you search with this thing called digital pathology or computational pathology, you would see this many literature. How many of them are in clinic? Maybe none of them, right? So the whole point is how we can do things together. And before I get there, I wanted to talk about a landmark paper. This is being published by someone from Memorial Sloan Kettering, whatever, that the New York hospital. So basically, they took all their slides, the scan, and they basically, it's published in Nature Medicine to classify different cancer types with high confidence, okay? And for that, they use 44,000 whole slide images. Do I have 44,000 whole slide images, or do I have space for that? Maybe not. I mean, it would a huge, gigantic amount of, data set, right? And then if we can bring the other type, it is just a histology data, right? Then you can bring all sort of spatial data and other thing. It's quite a bit of work, but they have shown this is possible, but they never talked about, right, disease progression, where maybe a patient is typically interested in what do I have is important, but what is going to happen with me is another important question, right? So even though we publish in Nature Medicine, is possibly the largest digital pathology repository, we are still not there to predict where the disease is going, correct? So I'm not saying like, we work on this or what is bad, what is good. I'm just trying to say this is the state, right? And hope here is that this Ph.D, this company, they got their first tool for prostate cancer detection or diagnosis being approved by FDA for using clinical use. So we have hope there. So they are leading the way, right? So that being said, let's come back to the topic of kidney and let's see, I mean, how at least we can make some case to go from simple level to make some convincing case to generate the trust in doctors. And here we can make it more and more, go more and more deeper, right? So what are the basic kidney compartments? I don't need to tell any of this to this group. So, right, kidney, maybe for the right? We have glomerulus in kidney, right? And then you get tubule. Then there are vessels like large arteries, arterioles, and capillaries, and interstitium. Either there is nothing, they're very hypothetical case, or they are right? So these are the structure. So question is, before even try to find pneumo in a tissue with all sort of molecular underpinning and find out if we can predict progression or not using that great machine learning algorithm, which could possibly lead to like world changing thing. Can we segment these structures, right? That's where I started when I started back in 2015 as a junior assistant professor. Because the whole idea was to get my first R1 for survival in medical school and to figure out the area where no one is working. So kidney is the NIDTK is the one who was looking for computational stuff, but people were fighting for funding in cancer. In digital pathology, this was wide open, so we started working on this. And first question we asked, forget about like whole slide image. If I'm given a crop like this, can I segment the glomerulus somewhere? Right? So we applied a technique. It's a very basic technique, like image analysis 101 for any graduate student. So basically, say this dog, how you can separate this out from the background, right? And there is the one thing you notice, the texture here is very different than the background. Texture means intensity change or frequency, right? But at the same time, so frequency change is something we call so-called frequency domain analysis, right? Not spatial analysis, domain analysis. So this frequency is very different than background, but while doing that, we need to preserve the boundary, right? So I will not go to the details of the math, but we figured out the texture of the glomerulus is very different than the background. And we published that, right? I mean, that time we also needed to publish, to get the first R1. But the next question came up, okay, if we can segment the glomerulus, right? Can we segment that or detect or segment that from the whole side image, right? So we use something called local binary pattern. That's also another texture finding technique. It's not very important what local binary pattern is. It's kind of texture maybe in a little more generalized way, right? So we also have shown that in a publication that we can use this technique to detect all these glomerulus, right? And apparently, I mean, that was not that difficult for us. I mean, there are tools and techniques available to segment these structures. But the question came up was, are all these were generalizable? Generalizable means concept of generalizability is very, very complicated. So question is, can this be used? And if I have a model, right, a trained model, right, which can detect all this, can I bring that, say that time I was in Buffalo, can I bring that to Florida and it would work, right, in your data? And I can very guarantee you, I mean, other people will say, oh, my model was everywhere. I would say, no, it will not work anywhere, right? Because it's in an image, right, in this tissue image, the biomedical data, before even forget about any machine learning or analysis, before even doing that, the few important things come up which you need to think about, right? And you do that always in your clinic, right? We also do that QC/QA, right, of the data, heterogeneity of the data, right, and the pre-analytics. Here, they can make the thing so different that always the models fail or they provide suboptimal result. So, but while we're working on this work, our other colleagues, interesting, this community is still small, like Aryadas is in Lithuania, Vijayakulla Chalmaskyov in Boston University. There are few people. They also talked about glomerular segmentation algorithms, right, using machine learning. But what we came up with later, using they use deep learning technique. Possibly you've heard about deep neural network or AI, right? Like large models, right? And what we came up with a new model, right, which can detect, right, all these glomerulus and their boundaries. And we published that in Nature Machine Intelligence. Apparently, I post about this work. It's in 2019. We did another Nature paper, right? So to post about it. But that being said, what we talked about, your model will never be generalized, right? What you have to do, you have to make a systematic way to interact with the computer when the results are generated, so that you can interact with those boundaries and you can correct that and get that to retrain in such a way so that you would not be spending that much time if you are training everything from scratch. Does it make sense? Means I bring my model to University of Florida. It will no way be optimal. It will be suboptimal. You run 100 images. You then kind of scan through them by eye. 5 of them will be bad. You correct them. You retrain the model. Okay. And that's how we have shown that it is far more efficient than training from the scratch. So, but I mean, that time the PR pressure was there. We need to possibly by that time, just by using glomerulus, I got my first R1. Just glomerular segmentation gave me first R1. But time to go beyond glomerulus, right? My first R1 apparently talked about just glomerular segmentation, feature extraction, disease prediction, everything based on that, right? But whenever we get the first R1, we think about the next one, right? Or how we can expand, right? So we talked about segmentation of this thing where the structure is not defined, which is called interstitial fibrosis and tubular atrophy. In this room, I don't think I have to explain to anyone what this interstitial fibrosis and tubular atrophy is. The interesting fact about this is, which I should still re-utter, that if I give one slide and put 10 pathologists in 10 rows to mark this boundary, all of them will come up with different answers. That's what for sure I have seen that. I never believed that, but when I saw that it's happening, oh my God, I see that they have like different, and it's so much. So we thought about, okay, train the computer, right? When you train the computer, the typical question doctors ask, how much of the biopsy is it? Typically, there are like 0 to 25%, 25 to 50, whatever, and then how much, you see, there are 4 discrete categories, right? 0 to 25, 25 to 50, 50 to 75, or 75 to 100, right? And how many of the glomeruli are sclerotic, right? They count that, right? So we developed this tool, which we published in JSM, right, in 2021, right, to show, right, we can reliably predict if the end sclerotic. But one question might come up from you, hey, okay, I mean, you can do that, but who is the ground truth? Because you said this disagree, right? So we basically, in this publication, we use the most senior pathologists who has more experience, right? But there are other ways to do that, right? Maybe we can image collagen in a very smart way. And this is the area where I'm working on, is a cutting edge new microscopy technique called dual mode emission and transmission microscopy, right? Where we image collagen, right? And that provides us the information of where the ethta is. And this whole technique, the beauty here is it can image it from histology side. You don't have to stain or unstain anything, right? We use this data to train the model to find out the fibrotic region. So that work is ongoing. This is the one RO1, but I'm a co-investigator with significant effort, but my colleagues in UC Davis, they are working on it. But again, I mean, I wanted to tell you that literature also, more like Vijay, again, his group also published on interstitial fibrosis and tubular atrophy. So, but again, you would see the same names are coming back and back again. So we're kind of working closely together. Speaking about ethta, how about PTC, right? I think everyone wants to know how they are related in AKI, CKD, inside the ethta region versus outside the ethta region, right? That's one of the quantitative measures you want to do. No, we did not publish it. Our colleagues published it. Laura Barisoni's group in Duke University. They recently have this paper came up, right, for peritubular capillary segmentation. Why it is important? For this audience, possibly I don't have to explain, but if this is my peritubular capillary, right? So if this is my peritubular capillary, right? And if this is the tubule, right? And if there are fibrosis, right? Imagine that how much path that molecule during the exchange has to cover, right? And that's why in a large data context, segmentation of PTC and understanding its relationship with the nearby tubule is so important. Maybe using big data context, when we try to solve finding Nemo problem, that could give us some clue, right? So that's why I find like this is intriguing. But what is more important that we also work with, apparently we work with possibly everyone in this domain. And we have their model available in our cloud tool that I wanted to show you. So in computational literature, it is also important that whatever you are developing in your lab, someone else can use that. And that type of reproducibility is called quote unquote fair by FAIR, by NIH. F means findable, A means accessible, I means interoperable, R means usable, right? Whatever someone else developed, can I use that? And I will show you an example that we can apparently read someone else's code in a standardized form. And we're also working on that, how we can standardize other people's code and ingest that all together in one platform, right? So that being said, peritubular capillary is there. So now it's time to go for multi-compartment segmentation, right? And we are very happy to say that, we can segment everything in kidney now these days. I mean, well, not everything, but reasonably in decent way, whatever is needed to start work, right? This paper just got accepted in Kidney 360. I was not so happy, right? Impact factor is still 3 or something. I mean, but what is intriguing is we are invited to be on the cover. It will come in the December issue. So you will see if you are reading Kidney 360, yes, we'll be on the cover. And while we talk about multi-compartment segmentation, our other colleagues, they also work on that. So there are 2 works by Anand Madhubhusi's lab, who was in Emory University, who is like the big pillar of digital pathology in our area. He works in cancer, but now he is building a large AI institute in Emory University. He recently transferred there. And Vanderlag's group, this is a group in Netherlands, right? He's also someone who works in digital pathology, but in kidney pathology. And they are starting a company these days and talking about some of the things that we do. But that being said, while people work on this, if you heard about something called end-to-end system, I would say that we are there, our group, and that's why next year I will not be writing any R1 grant. I will be submitting few SBIR grants to see, including Arcana Labs, to test the ability, stress test our tools using all the biopsies for Arcana Labs to see, if you heard about Chris Larson, right? So we work with him to figure out, what we can do with that big cohort. And hopefully my system will not fail. And I will show you our end-to-end system. And this end-to-end system is here. We have developed using our hypergator system, where you can see, right, for example, What is my, for example, here, not Colorado, it's going to be Colorado. So this is a digital whole slide image posted in cloud. Right now, this is in Hypergator in a location which is public facing. We are working on so it public facing means UF is very strict about security. We know our IT, right? How strict they are. For a single dictionary, they can be a part-time English dictionary. So even for a single English dictionary, IT is coming after me. So imagine that if I want to make this public facing for using all over the world, where people will come here and use this tool, how much level of work I have to go through. Apparently, I found that a pain in the beginning when I was coming, and I just didn't like that. But now I realize, even though you don't like them, there are few people there, and you actually learn out of that how to be more secure. And we are actually doing that. We have a big software unit in our lab. We are working with IT, main government IT, to do this. So right now, this is public facing. This has limited ability, but we are bringing. We are using all the capability of Hypergator, which will have all security in place, which will go via UF login ID and password, right? That should be replaced. But what you can see, like this image, this is posted via cloud. We have an analysis button. It looks a little ugly. We'll change that up very soon, but you can see this segmentation. We have all these different models, right? Multi-compartment, PTC segmentation is coming from Laura Barisoni, right? So when you run this multi-compartment, right? I will not run this, but I wanted to show you the type of model file we have, right? So what is the model file? Right? So you have these many different models, right? So you have like PTC segmentation is a model, and that's available from someone else. Then including our model. In glomeruli model, you see like all different models. Someone did some competition, that's from their model. We're bringing everything in a standardized form from here so that people can use that for better usability, to create a community where this can be used, right? And this paper will be published very soon. Right now, I'm not run this, but you can see like once someone hit and submit, they should be able to segment tubules like this. Whatever I'm doing that, by the way, I'm not running anything. This is the result I'm showing, like all the tubule, right? Globally sclerotic glomeruli, right? Then non-globally sclerotic glomeruli, all peritubular capillaries, right? All peritubular capillaries. There are some false ones, but and then IFTA, right? So now you get IFTA is actually this one, right? That is the same color. But now what we are also doing, we have a feature analysis button, right? So feature analysis is this one, feature extraction. So you can imagine that you can take this image and you can extract features from there. I will not run that, but I will show you how that one looks like. So if I go here, right? Right, it's from someone's account. So it looks like this. So from an image, after all this segmentation, you can get, for example, all glomerulus, they are coordinate, but their information like their area, their mesangial area, mesangial fraction, all sclerotic glomerulus information, all tubular information. You can imagine however many tubules you can have that. Instead of looking at few tubules, you have all this data like their TBM, average TBM thickness, right? Average cell thickness, like epithelial cell, that region thickness, luminal fraction, arteries and arterioles, and all those information. So you can get all those, right? And you can download that, right? And then you can use that for your secondary use. Maybe to compare with other data, like other orthogonal data type, right? So that being said, I would just given all this, right? This system is available for end users, right? So always a so what question, right? And the question is, can I bring that trustability in doctor, right, using our tool? And we published this work on diabetic nephropathy classification. You know how the diabetic nephropathy classification versus via based on RPS system works, right? People look at all these glomerulars and based on the most worst one, right? They provide some sort of grading. Of course, people also look at if and other thing, but that those things were added later. But what we developed, right, by looking at the glomerulus, right now, because you have seen, we can take a outside image in an end-to-end pipeline, right? We can segment all the glomerulus, we can extract their features. We input those features in a machine learning model to find out, this staging. And what we have done, we looked at those staging, how reliable those are with respect to pathological grading, and we found we got comparable results. For the sake of time, I will not completely explain like all this data, but all of it say that we kind of concluded that we can get as reliable as what doctors stage. And this was by Brandon Genley. It's kind of considered like as a seminal work using AI for classification, at least in kidney literature. We got huge amount of citation on that. It's published in 2019. Then another so what thing is that how we can bring this tool for kidney precision medicine project and how we can make an impact. So all of you who don't know what kidney precision medicine project is, so basically kidney precision medicine project, it's a multi-year investment by NIDDK to understand the kidney, right, better for AKI and CKD patients. The patients are recruited by the consortium by several recruitment sites. We are not one recruitment site. So by several universities, once those kidney data is collected using some sort of protocol with other data, including urine data, serum data, and serum measurement, and other clinical data, they go to a central hub, which is University of Washington, led by University of Washington. They go to their archival, and from there, this data is distributed to several tissue interrogation sites. Tissue interrogation sites are from different universities. Those are U grants led by people who are using different molecular or spatial technique to interrogate that tissue to find out if we have a new information, molecular information with the data, which upon learning, we can paint that back in an image using some sort of immunohistochemistry technique to understand or use something better than what the current clinical practice can do. So that's the overall goal for KPMP, right? And we are focusing on understanding CKD, understanding AKI, progression from AKI to CKD. As I said, this is the continuum, and I am the co-lead, right, for the image analysis for this whole consortium. So that is one thing, right? And the goal, if I can, explain, right? We have the histology image, and by now you have seen this segmentation and feature extraction. But can we also, right, correlate this data with some sort of orthogonal molecular data and talk about this new area called so-called pathomics, right? So the whole idea is here that integrate all these different kind of data and stratify patients, find out, differentiate healthy tubule like from happy tubule from unhappy tubule to establish that continuum using KPMP. And by 2027, we are hoping to recruit 1,000 patients. By now, I think there are more than 200 patients already recruited. And it's pretty fast because the first patient was recruited after all protocol and everything was done in 2019. So these are the application of our method in different institutes data using different protocols where we are showing the versatility of our tool. And not only that, this is Nick, possibly you have seen, right? He is a student in biomedical engineering department, right? He is possibly the one, I mean, while people don't know much here, but he is known by all KPMP because he just go to the state podium with me to present our data and what we are releasing for the community, right? And one beauty of KPMP is the patients are present also there. So if you go to the KPMP website, you will search by patient. I will see like we work directly with patients to see whether, what they are thinking, how they are interpreting that. Because at the end of the day, imagine that if we can find Nemo in the histology, right? You finally trust, okay, we can use this tool for some sort of analysis. You have to also convince your patient that I am using AI for your analysis, right? Because all protocols are there, right? So we have to bring patients in this. So what we have done so far, this data, and for this, data we have in KPMP, we following all our technique, we extracted this feature for all this compartment, and they are released via KPMP address. So the image data, molecular data, and extracted feature data by us are available, and it's available for anyone, right? People can use that to study those data. And even you can compare that data with your case. If it makes a hit, right, with certain features with molecular data, right, you can also come and look at in your data and see what you are saying. Right? Those are the opportunities for the entire world that we are creating using KPMP consortium, and we're also working with other groups. So Dr. Gehlenbach, he is kind of world renowned for data visualization. So as part of the KPMP, we are also working on how we can bring the results of our tool to a useful visualization system, which users can use using all sort of button to understand, different feature amendments, right? So there are enormous possibilities, and it's not too late for someone, to get involved in KPMP. So another what question is outcome trajectory based. That could be of interest. And I would show a very pilot study, small study on that, because I always get so much engaged with the current work I have. We didn't get much opportunity to do the outcome prediction work a little, but our current grants are all require us to do that. So this study, right, from 29 transplant surveillance biopsy cases, and we extracted all their glomerulars, we extracted the glomerular features, and these are each dot is a glomerular based on that feature, and they are plotted in some sort of a 2D human plot using dimensional reduction. And these are the different clusters that popped up when we did clustering in that data. Now, if we level these dots, right, based on where these glomeruli are coming from, either EGR dependent, less than 5 per year versus greater than 5 per year, we found that, this is the, severe one, which is kind of matching with this, this cluster, which tells us that this cluster has, different, interluminal distance is larger, internuclear distance is larger, large glomerular area or PS positive area, which are so-called, digital feature or so-called so for finding Nemo problem, or toward that finding Nemo problem that we can do. So that being said, this type of feature analysis, right, going beyond what is there in current clinic, is also being pushed forward by our other colleague, Peter Boer's group, right? His landmark paper came in Nature Communication. It's a great paper, someone can read. They're also talking about that. And he is a large cohort of data of diabetic nephropathy, lupus nephritis, IgA nephropathy, has shown this continuum, right, using their cohort in, disease trajectory prediction. So what I wanted to say is that there is no competition here in between different groups, but we're all working together possibly toward the same vision and trying to establish this area of computational pathology. How about detection of single cells, say podocytes? Again, I would go little first, but podocytes. So we have published this work, like detection of podocytes from histology image. It says label, but it should be label free, because you give an histology image in the input and it spits out all the podocytes, and it came to the cover of JSN. Maybe little believable project would be labeled one. It reads label free, but it should be labeled, which is called podo count, which we published, where we have shown that we can detect all the podocytes from immunohistochemically stained podocyte images, right? But for us, it's a pretty simple project. But we also have published that. But what I found out of these two projects, the second one got more impact because it was used in various mouse studies, right? Where I'm a little, author in all of them, right? Here in all of them. So we're expanding that in use of different mouse model studies where we contribute of this, podocyte detection and feature extraction and finding the correlation with other set of data, urine data or molecular data, and these are the impact we are seeing. So scaling up, can we predict or paint more cells, right? Not single podocyte. So I am very fortunate to be part of this consortium called Human Biomolecular Project Consortium. So let me tell you what that consortium is. If you can go to their website. So the goal of this consortium is to model the whole human body at single cell, even single molecular level, right? Because there are trillions of cells and generate a knowledge atlas, right? That could help you understand better the human body. And what we are doing in that consortium is studying reference tissue, not the disease tissue, but reference tissue, because learning at big scale, it would so enormous, disease would be very difficult, right? So we are working on a project here, which I call Operation Nebula project, which is beyond imaginable, where we are saying, we're asking the question, can I bring a histology image? Can I use a machine learning model using that histology image? And can I spit out in the output all possible cell type? Not only all possible cell type, but what are their state? Are they in the regular state or degenerative state or cyclic state or whatever, like adaptive state? Can we do that? This is very, very difficult. It's beyond imaginable work, like just an histology image. Can we do that? All I can say that, well, before I tell that, I'm like, there are different groups. So I'm a PI of a group who are working on this tool development project while also contributing community tool development, and our group has multi-university grant. We have people from Nebraska, we have usability expert, because at the end of the day, someone has to use it, right? And Laura Badison is a pathologist, and Jessica is a AI usability expert, right? So we are very soon going to do a usability study of our work, like in January. Very soon that is going to happen, and anyone here is more than welcome to join. We have a data generation group. PDP means, it's like, teaching, undergrads, being underrepresented community to this area. And we also have a software team. So the data we are generating, right, is this data generation group, the high-end data they are giving us, like same-sex and histology and codex data, right? So codex is basically protein marking technology. It can mark up to 40 markers in one channel. And we are getting this data from same-sex with a goal that we can use machine learning to train this model using this as a ground truth and find out by this, using this input alone, to what type of markers are there, right? So that's the whole idea here. And for that, remember that we need to have a clean data. And that group we are talking about, they have O'Brien Center, so they're possibly the best one in generating this data. And we're also having adjacent section spatial transcriptomics data, which gets the transcriptomics information resolved in space to help us to train our model to about the disease state, right? Cell state, right? So where we are, so the pipeline is, this is the same section histology and codex data. This is the ground truth we generate from here using this codex data. So this is a ground truth of different cell types. This would take a 1 hour of talk on how to generate a ground truth, but I have a SF2 UF student in my lab who actually worked on that, and he is going for a platform presentation in February in a digital pathology conference in San Diego. And this is our preliminary results, and all we can say that right now, this slide needs to be a little modified. We are seeing the limit of our model. We can find out like about 12 cell types with 0.7 accuracy. Okay, 0.7 accuracy. The baseline is about 0.06 or something, but imagine that if I have to publish this in Nature, I need like 0.8 or something. So we're still pushing, hopefully. And when we do that, we're also increasing the data quality generation. And we are also trying to see, can we push that for other organs? And the organ data that we have in hand is skin, intestine, and lungs. Because you can imagine certain cell types, right? Injured cells, neuronal cells, right? Immune cells, injured cells, right? Or few other cell types, right? They are kind of common in different organs. So question you are asking, even a most difficult question we can ask here. can we train in one organ and test in another organ, right? So all these answers, hopefully, if I give the grand round next year, we'll know whether we can do that or not. And that thing is going on. And again, Nicholas, he hasn't passed his qualifying exam, by the way, yet. And he just transferred with me and he's working on this. So possibly he should give a talk here in the audience, and we'll see how that works. But maybe this is the time for developing a digital atlas of kidney. And when you talk about digital atlas, right, we talked about the level of complexity, which starts from a structure with the number of cell types present, with abnormality to diagnosis, to quantifying the abnormality, and to see what does it mean for a patient, and what the plan look like for bringing this to a clinic. And that's called the atlas, right? That's could be the atlas. And we are possibly going to release the atlas, like a digital version of that in a limited data set. And I hope that you will have a little time to see that. We are still, it's being kind of released to the outside world. I am working on a promotional video on this. And we are very soon going to look for users. And that's why I ask you, how many of you really use like molecular data, right? And when he says depends, that's the answer. Because when I talk about usability, right? If I'm doing something fancy, great. I mean, we talk about it. But if you're not using this, right? It's meaningless, right? So we are trying to figure out what can be used, what cannot be used. What it does, we have a system where you can do visualization of data with those structures and molecular information. It helps you to build the data set, because we have about 54 data set we are releasing with histology and molecular data. You can bring your own data set to do all sort of computation. How the visualization looks like, hopefully this will work. And as you can see that it's taking time to render, but I think property. So you can see, like, it's basically showing you the overlay of information, and then if I go to small muscle cell, when I say thick ascending link, these are the structures in the kidney, and they are abundant of thick ascending link with some number, okay, with some heat map. What this knowledge base does, not only it does that, it can also tell you the cell composition, right, in this area, right? Different cell composition for glomerulus, tibial, arterioles, and not only the cell composition, but their state, altered, degenerated, reference state. And you can also select a region here. Imagine that you have a diabetes tissue and you want to know the cell composition and state from fibrosis to non-fibrosis. There are several things which are unknown to us. We are trying to understand that. We are bringing that to people so that they can do research, right? Because I am not a cell biologist or by training, right? That kind of research someone like you can do, right? And then we have also cell graphics, right? We are thinking about like a digitizing textbook, right? So like this is kidney, right? So ideally you would select in all of them and it would give you some sort of cell graphics, which is not here yet, but at least the cell hierarchy part is created, right? So you change it and it shows the different cells. Let me see which one has a bigger hierarchy. For example, it tells you like for that particular cell, all possible ontology, right? Like from organ level to cell level to what type of molecule they express, to what type of antigen, antibody marker that you can use. And the vision is you click all of them and it will give you that information or a Wikipedia, which you can use, right? And when I say the tools, what is fascinating is morphological clustering. For example, right, there are a bunch of slides, right? And say I want to see in a bunch of slides, right, morphologic information, right? For example, object area, total object area, right? Or with respect to slide, for hopefully this will work. It just didn't work few moment ago. So filter levels in yes. I'm just calculating this for moment. It's not very, let's see. Here you go. So it takes bunch of slides and it calculates their difference. So basically you can bring your data and it can show you the difference. Not only it can show you, so basically there are two slides now, all the glomerulus and their features, and it's showing their difference. Okay. So basically you can take bunch of diabetes slide, bunch of normal slide, and you can see the difference. Not only you can do that, you can click on that and you can see the image. Okay. You can not only you can see the image, but you can see their cell composition and cell state. And you can imagine that this tool we are building, I'm creating a promotional video to sell this, not exactly sell this is all open source, right? I realize that in university setup, if you have to think about a company, the amount of pain they give us with the conflict, better not do that, right? So we're not going to sell that. And as you can see, the level of complexity it has from where we started, from segmentation to feature extraction to establishing a new domain to molecular level, where Asutosh says it depends. So where is the usability? I am working with the usability team to define the usability, and real nephrologist, pathologist team, they are working with us with usability expert. In January, we'll be ready to do the usability. Hopefully we can recruit some of you, right? At least you'll be in the papers. And again, this tool, the way we envision that this will be for everyone, including patients, right? That's where we are going, right? It's time to wrap up. So I think I was a bit quick, like, when I first came in US in 2003, people said you speak too fast. It's kind of Indian thing, but the problem is today I was late like 10 minutes, right? But I was still able to finish it just by speaking really, really fast. Hopefully you understood what I was saying, but I hope I was able to tell you what is digital and computational pathology, where the field is, how the field is shaping up with not only by us, but with our colleagues, where it is going, and what the state of the art would look like, and opportunity. Yes, the opportunity is there. I'm very fortunate to have PI R1 brand. I have a R1 brand in diabetic nephropathy. I am a PI of a R1 brand in transplant. I have the one where the award letter is getting prepared on lupus nephritis. I'm part of KPMP. I work in HubMap. Only R1 brand I'm missing in my lab is on IgA nephropathy. If we get that, everything in kidney will be made by our team. So but I don't have time for now. So if one of you wants to write it, you're more than welcome to join us. It will be your brand. You can be the PI. It can be a small PA or something. But possibly you can write better with enormous resources that I'm bringing this department, right? For IgA nephropathy. So that being said, thank you very much.
Great work. So you're taking the slide. You first, at the beginning of the talk, you said the tremendous amount of work to go from the renal biopsy to the pathologist. Now you've gotten sort of eliminated the pathologist perhaps, and you're getting down to the cellular level, which can provide insights into the pathogenesis of disease,
yeah,
and the future, much predictive, getting into the prediction for the patient. But what about at the biopsy? Is there ways that you can just take a biopsy without being scanned, without special stains, without being segmented, and look at it and predict and make a diagnosis without all of this special stuff, by just doing different fluorescence and how it reacts and absorbs and everything else?
This is a great point. Great minds think alike. So if you're thinking that, maybe someone else is also thinking that. So our colleagues in KPMP is working on that. They are using autofluorescence, right? No staining, just autofluorescence. Other type of staining to look. They are investigating the limit of what they can do or they cannot do. And we are working with them. As I said, that my goal is not to just establish us, but to moving the field forward So we are also working with them to see and bringing their tool in our cloud system, I said. His name is Jeffrey Spraggins. He is in Vanderbilt. His team is working. But yes, I mean, there are works, but again, if you ask about the intricate detail, like what we can do, or there are limits, definitely, there are certain things you can do versus, so that part he is investigating.